There is a chat box at the very bottom of your screen. So if you kind of hover over the bottom, you'll see a chat. Please feel free to write your any questions that you have in there. We want to make sure that your questions are answered tonight. Me and Dr. Greenberg have spoken a few times, and you know, the goal of this webinar is to get some answers to educate you. And you know, there's been a lot of you know, kind of hype and a lot of questions about SRT and really understanding what that means. And that's the goal of tonight, is for you to understand and walk away with you know, a really good, clear idea of what that means. So thank you for joining us tonight. Dr. Greenberg, especially you, because thank you for your time, and especially knowing especially knowing that you're with your family and your grandchildren. It really means a lot that you're here. And you know, you are a huge part of our community. For those of you who don't know Dr. Greenberg, she's actually helped us out with some of our patient conferences as well. And you know, she's a great part of our
click on launch meeting. Probably already done that.
Yes. So anyone that's not muted right now, I'm just going to ask you to, yeah, at the very bottom of your screen on the left, you're going to see mute. Feel free to leave your videos on if you like. That way we just don't have any feedback for when Dr. Greenberg is talking. And without further ado, Dr. Greenberg, we can get started. So I'll share my screen with you, and I'll make you the host. And for those of you who have any questions, again, please feel free to write them in the chat box. And we're going to have a 15-minute question period at the end as well. So you'll have some time to get those questions answered.
How's that?
That is good. And Vera, if you don't mind just muting your microphone. It will just be, I was going to do, yeah, thank you so much. Perfect. And anyone else that just joined, I think that we're good. Okay, so thank you so much. And Dr. Greenberg, I'll hand it over to you.
Okay, thank you so much, Julia. And hi, everybody. I know everybody's in different time zones. That's why on my screen with my image, you can see the background of a typical Winnipeg sunset with the Canadian Museum of Human Rights in the background. And I'm really, really honored to speak with you tonight. You know, whenever you prepare for a webinar, you learn so much. And I've had the opportunity to speak with people who've helped me, and you'll see in my acknowledgements, really provided me virtually with all the slides that I'm using tonight, even though I've been intimately involved in Fabry disease for many years. It was a good opportunity for me to try to dig a little more deeply into what some of the issues are. And Julia asked me to talk about substrate reduction therapy in Fabry disease, and I'm very pleased to do so. In Winnipeg, whenever you give a talk in a public domain, I always do say a land acknowledgement, particularly when there's people from all over Canada, and we acknowledge that we are gathering on the original lands and traditional lands of which we all reside in Canada. We respect the treaties have been made with Indigenous people, and we acknowledge the harms and mistakes of the past, and we dedicate ourselves to moving forward in partnership with Indigenous communities in the spirit of reconciliation and collaboration. These are my disclosures. I know there are many people from industry on this webinar, and I do acknowledge all the support and help you've given me over the years. I hope after this talk that you'll be able to easily describe the signs and symptoms of Fabry disease. I know many of you just live with them every day, but to actually articulate what that means in a scientific forum becomes sometimes a little challenging. And I want to make sure you understand the serious complications of Fabry disease, and two new different approaches to treatment. This is besides enzyme replacement therapy. And be able to understand the pros and the cons of these two different approaches. And of course, I didn't put this as an objective, but I'll leave you with, you know, I hope what you will feel will be lots of hope and excitement for the future. So I'm a metabolic geneticist. I deal with taking care of children and adults and their families with inherited metabolic disorders, what we used to call inborn errors of metabolism. And, you know, I think you all know we have about 23,000 pairs of genes in our genetic makeup. And there's about 700 known genetic disorders where a misprint in one single gene can cause a serious disorder. Of these single gene disorders, there's probably over 700 that are now classified as inborn errors of metabolism or inherited metabolic disorders. And these are genetic disorders that they're caused by an abnormality in either the quantity or in the function and the structure of a specific enzyme or the protein. And that's what I show in this box here. Fabry disease is caused by a faulty enzyme, and enzymes work on molecules that we call substrates, and they convert them into a product. And if you have a faulty enzyme, the implications of what kinds of signs and symptoms it might cause really depends on what's the effect of the accumulating substrate. The enzyme cannot act on this molecule. The substrates elevate in concentration, and what kind of toxic effect do they have. But also we have to keep in mind then when there's a faulty enzyme, we're not making the product that is needed from this enzyme reaction. So the signs and symptoms that you might see in inborn error metabolism are usually a combination of whether the defect is a quantitative one, a qualitative one, whether the toxicity is related to the accumulation of a substrate or the deficiency of a product. And very often it's a combination of things, and there's also many factors that we don't know about. So I've been in Winnipeg as a metabolic geneticist for 42 years, and
So Dr. Greenberg said her internet was acting up earlier, so I hope that she's able to come back to us shortly. We'll give her another couple minutes here. It looks like she may have dropped off, so maybe she's coming back. For the patients that are on the call, are on the webinar tonight, we can maybe be a little interactive, and you can flip on your video. You can flip on your video or your microphones. Has anyone heard of SRT before?
I've heard of it, but I'm not really very accurate in terms of what it actually means.
Mm-hmm. Yeah, so it's definitely a new therapy that's that's around, and a word that we'll see more and more of for sure. And two companies are working on SRT right now. Oh, I see that she's back, so that's good.
I'm very sorry. I get
That's
to start
okay.
my video. Okay. All right. I'll be penalized for the time that I left you. Now I have to share again, huh?
Yes.
Okay. So I was just talking how we approach clinical problems. Did I lose again?
No, you're just not shared.
I'm not shared. Hold on a minute.
Disabled participant sheen screen.
You have to share my, let me share my screen again,
Yes.
Julia. Sorry.
No, that's okay. How about now?
Okay, is that okay?
That's great. Perfect.
All right. So I was saying that it was almost like a circle, how we approach the clinical problem with our group of clinicians and basic scientists to try to solve a problem. But in reality, what it looks more in terms of how clinician scientists view, like myself, view the world, is that research has to start at a basic level. And instead of seeing a circle, we're really dealing with hills and valleys. And there's often a valley after we acquire basic knowledge. And then it's a situation of applying the basic knowledge into a clinical situation. And that's what I'm going to talk about is clinical trials. And once we have identified and have the results of clinical trials, then it's the last major hurdle to cross this valley and be able to implement health decision-making and making sure that the treatments that have been proven through clinical trials actually get to the patient. And that's a huge hurdle, but we're going to focus on the clinical trials. Now, just for the sake of time, I won't spend too much time because I've already lost some time. Many of you know that Fabry disease is caused by the deficiency of the activity of the enzyme alpha-galactosidase. This enzyme is encoded by a gene on the X chromosome. So this is an X-linked disorder. And the deficiency of this enzyme causes accumulation of the substrate before the enzyme block, the buildup of what we call a complex lipid called GL3. And it's this excess lipid and some of its other molecules similar to it that cause cell damage, that ultimately lead to tissue damage, and untreated leads to organ damage, and untreated can lead to premature death. And I'm going to talk about the advances in treatment. First, some definitions. Classical Fabry disease, in classical Fabry's enzyme activity is usually measured less than 5% of expected. Often it's less than 1% of alpha-gal activity. In classical Fabry disease, more than one system is involved, marked down here, and the manifestations involve many systems and are diffuse and often very, very debilitating, as you know. In non-classical Fabry disease, there's more residual enzyme activity, and you would expect. Usually just one system is involved. Often patients lack the pain, angiokeratomas on the skin, and other symptoms. The most common problem in the non-classical or cardiac variant of Fabry disease is in fact heart disease, but it can involve other systems. The age of onset is variable, but can vary from late adolescence to the 70s. It still can have potentially life-threatening disease, for sure. And it's much more likely to be missed than classical Fabry disease, although there is still an unacceptable long delay in establishing the diagnosis of both classical and non-classical. In some cases, in non-classical Fabries, you might need an invasive procedure, such as a biopsy, to make the diagnosis. Many of you are familiar with the morbidity associated with the cardiac manifestations of Fabry disease, whether you're women or men. As you can see, a high proportion have serious complications related to the heart, and often it's the first serious complication of Fabry disease. The age of onset of serious cardiac complications is younger in males than females, but I don't distinguish in classical Fabry disease. Females have similar morbidity in many cases than men. And overall, well, more than half of adult patients will have some evidence of heart disease. For the longest time when I arrived in Winnipeg, we did not have patients identified with Fabry disease, even though we looked in lots of clinics dealing with the end organ involvement, renal failure, cardiac hypertrophy, but because we knew that a proportion of these patients would have underlying Fabry disease, albeit it's still a small but significant important cause of unexplained hypertrophic cardiomyopathy. The same thing with kidney disease. This is often the most serious life-threatening complication of classical Fabry disease, and approximately 2/3 of untreated men have this. Women can also have renal disease, but not to the same level, although 10% may still have renal failure. From the point of view of the cerebral vascular disease, you can see that strokes are higher in patients with Fabry disease, and you can see the average age of a first stroke is much lower than in the general population, where there may be other risk factors. And you can see the risk for stroke is much higher than the age-matched general population risk for stroke. It's about 12 times as high. Many patients suffer GI disease. I think most of you in the audience will have experienced that if you have Fabry disease, although sometimes it's hard to know because abdominal pain is so common in the general population. But the malaise from diarrhea, vomiting, and nausea is very significant in patients with Fabry disease. Probably the one that's most debilitating for so many individuals is pain. And this deserves a slide of its own, because pain is often the first symptom and it can be very, very debilitating. I want to make sure that I have a slide about pediatric patients, because in dealing with clinical trials, very often we ignore the pediatric age group. But as a pediatrician, I'm a pediatrician also, it's important to include pediatric patients in clinical trials, because children are not just small adults, and they certainly suffer morbidity of Fabry disease and present really quite early and with signs and symptoms. So clinical trials have to also address the needs of pediatric patients with Fabry disease. And you can see often pain is one of the most debilitating. So early in the 90s, enzyme replacement therapy, but replacing the missing enzyme, was introduced for the treatment of the lysosomal storage disease, which is a type of LSD, as Fabry's is, called Gaucher disease. And that was introduced in 1991. And I'm not going to talk about enzyme replacement therapy. And before that, perhaps many patients had to have stem cell transplants or organ transplants, which still, of course, can happen. I'm going to talk about substrate reduction therapy. I'm not going to talk about chaperone therapy or enzyme enhancement therapy. And I know there's much exciting work that's being done on gene therapy. Again, I'm back to my discovery to practice slide, and I'm going to be dealing again with the clinical trials of substrate reduction therapy. But first, I want to make sure that people understand what a clinical trial is. Clinical trials are a way of looking for new ways to prevent, detect, control, or treat a disease. You have to do clinical trials to be able to develop and accumulate the evidence that is going to allow you to prove that a new drug is both safe and effective in treating a problem. And when you do clinical trials, it's a very, very complicated process. I've learned much from many people who are very experienced in developing clinical trials, in understanding methodology, in understanding how you set them up and how you evaluate the data. And there's so many different aspects of clinical trials that could be addressed in this kind of talk. But it's important and as we're talking about today, clinical trials of substrate reduction therapy modalities for Fabry disease. When you organize a clinical trial, it's really important to try to include many different types of peoples in clinical trials. I mentioned the importance of including children, but they're often not included till after the initial trials are done with the adults. Important to include people with different ages, ethnicity, races, and genders to see how different people respond to a treatment. You cannot sometimes go from results from one population and be able to say exactly what's in another population. And the clinicaltrials.gov website lists several thousand trials that for so many different disorders that are currently underway. So what do we know? What do we know about Fabry disease? Well, this is the enzyme alpha-gal A that is important for degrading a complex lipid called GB3 into a compound called sphingosine, which then is used in other aspects of glycolipid metabolism. At the same time, a precursor molecule called a fatty substance called ceramide is used in a series of steps to synthesize GB3 with the intermediary glucosyl ceramide. And this is the biosynthetic arm of how you make GB3, and this is how you degrade it. And enzyme replacement therapy are the modalities that replace, giving the exciting advances in ERT and human recombinant technology, replacing the missing enzyme so that this molecule does not accumulate. But I'm going to talk about the molecules that inhibit this GCS enzyme so you don't make too much GB3 because you have problems degrading it in Fabry disease. So one targets lessening the biosynthesis of GB3, which is offending substance that accumulates in fabrics, and the ERT and chaperones are needed to enhance the degradation of GB3. So the chemistry of the compounds that I'm going to talk about today, lucerastat, venglustat, these are what are called immunosugars. There is one on the market, eliglustat, that's actually, it's also a compound that is manufactured by Sanofi Genzyme, and it is an oral substrate reduction now for the treatment of Gaucher disease. But I'm going to be talking about these two substrate reduction therapy compounds, lucerastat and venglustat, for the treatment of Fabry disease. Now, I wanted to try to keep my slides really simple. It is really hard to understand, but you know, the previous one was a real good cartoon, but I just wanted to show you in real life. This is that GL3 molecule that accumulates in Fabry disease because you're missing this enzyme. And all the degradative molecules that have to that don't get formed, this molecule gets accumulated. It's toxic. It gets converted to another toxic compound called lyso-GB3. But if you can prevent the synthesis of this compound by inhibiting glucosylceramide synthase, this GCS, by substrate reduction therapy, the two compounds of venglustat, serostat, are the investigational products that are very promising. So what do we know about lucerastat in Fabry disease? How does lucerastat work? Again, both these drugs are investigational drugs. Lucerastat is manufactured and is being studied by Idorsia Pharmaceuticals with head office in Switzerland. And again, it targets the accumulation of GB3. Its mechanism of action is substrate reduction therapy. It inhibits this enzyme, glucosylceramide synthase. So you balance the production of GB3 versus the elimination. Reduce the production, and therefore the net effect is to lower the accumulating GB3 and its other byproducts. So whereas enzyme replacement therapy sometimes has its limitations, if we have time we can talk about it, substrate reduction therapy is being investigated, has the potential to treat all Fabry disease patients in the sense, regardless of the type of mutation they have in their alpha-gal gene. It's for the treatment of male and female, for children and adults, although the data on children have not yet started because clinical trials in children have not yet started. It's an oral monotherapy, a single drug that's given orally twice a day, versus enzyme replacement therapy, which is given intravenously. And again, if this is the enzyme that is blocked in Fabry disease, enzyme replacement therapy will replace it. Chaperone therapy will give a little kick to whatever enzyme is functioning, residual enzyme is functioning, and that can help the degradation of GB3 so it doesn't accumulate. In lucerastat is that molecule that inhibits the GCS enzyme and lowers the synthesis of glucosylceramide and GB3. So this is another cartoon that really is trying to show the balance between production and elimination, and how oral lucerastat can help reduce the amount of GB3 that accumulates when there's a missing enzyme in Fabry disease. So what do we know about lucerastat? I mean, as I said, Idorsia has been studying this molecule, having developed this molecule, and has been studying it for quite a long time. Some of the preliminary work, sorry about that, showed that it requires twice daily dosing. It can be taken with or without food. There's low potential for drug-drug interaction. It can reach most body systems, and different than enzyme replacement therapy, the SRT molecules that we're talking about today can cross the blood-brain barrier, and both in animals and in humans, it can be shown in dorsal root ganglia and in the brain. And it's excreted in the urine, pretty much unchanged. So it doesn't require much metabolism. But you have to, when you're giving lucerastat as an oral agent, it has to be adjusted if there is renal impairment, which is what we do for the patients who are in our clinical trial. And from what we knew from preliminary work, there was no effect of the lucerastat clinically relevant on the ECG parameters. So I could only present to you the study that was completed, the initial exploratory study with Fabry's patients that was conducted by Idorsia. It's called the phase 1B. And it was to, basically, this study was a preliminary study. And it showed that when you added lucerastat to enzyme replacement therapy, you actually enhanced the rapid and additional reduction in GB3. It was well tolerated, it was safe. It is a proof of principle in terms of the mechanism of action because it significantly reduced the amounts of GB3 that were accumulating in the patients that you would expect, and other relevant biomarkers. And this was a relatively short, small study, but the preliminary results from the phase 1B study led to the development of a protocol, that I'm going to talk about in another minute. But in the 12 weeks of this study, it was safe, it was well tolerated. There were no severe treatment emergent adverse events that were felt to be directly related to the drug. There were all, of course, on clinical trials, meticulous reporting of all adverse events, serious and non-serious. And there was really in the initial study, exploratory study, the very good safety profile. So our center, along with others that I'll show you in Canada, became involved as sites for the study of lucerastat in a phase 3 study. And this is one that we're currently involved in. It's a prospective, multi-center, double-blind, randomized. These are all words you have to know if you're doing clinical trials.
It looks like she lost her connection, I think. She's still on here though, so we'll just hang tight and see. Does anyone have any questions as of right now? Okay, so she's left, so I'm sure she'll come back. Anyone have any questions as of right now? Is this are you finding it easy to understand, confusing? A little bit of both.
And so far, so good here.
Yeah.
Yep, that's good.
Yeah. It's
sorry for the light behind me, but
that's okay. It's exciting. It's exciting to have to learn more about this, and to have Idorsia and Genzyme as well. Working on SRT.
yeah, definitely. Oh.
Yeah, I agree. Oh, there we go. Okay, so she's back. I'll make you the host again. That's okay.
I'm so sorry.
It's okay. It happens.
I don't know why. This is such a tech house.
Yeah.
Can you let me share the screen again?
Yes, just did.
Okay.
Which one?
Which is it here? I don't know. Nope. I don't see my screen.
Oh, it's coming. We can see what you're looking at.
Your slides?
My slides, I don't know if the slides have opened or
You
not.
close them.
Perfect.
Okay. Okay.
Hey, Bobby.
Hi. Where was I? Can you hear me?
Yep, we can hear you.
Okay, so we're talking about the phase 3 program. So it's called Modify. There's an initial screening period followed by a randomization 2-to-1 to where you have 2 chances for the first 6 months of obtaining the drug versus a placebo. And then it's after the 6-month period of treatment. Of course, as a double-blind period, as an investigator, I do not know what treatment the patient is on, and the patient does not know, nor does the pharmacist or anybody involved in the clinical trial. And this is followed by an open label extension that's now actually been extended actually for an additional 2 years. And there's lots of monitoring and lots of follow-up throughout the clinical study. To take part in Modify, male and female patients must have confirmed Fabry disease. It doesn't matter what kind of mutation you have, which is one of the prerequisites for a chaperone therapy. You have to suffer from neuropathic pain. And really, this is one of the main outcome measures. You have to have significant pain as recorded on your diary during the screening period. At the moment, you have to be over 18 years of age, and you have to agree to complete the electronic diary for the first 6 weeks. And you can be, if you've been on enzyme replacement therapy before, you have to be prepared to stop it for 6 weeks and during the screening period, and 6 months during the treatment trial period. And then, of course, you are on the open label extension if you want to continue. If you're treatment naive, you can begin the modified trial right away. One of the things, if you've enrolled and are eligible for the modified trial, you can, if the discretion of the physician treating you want to add back enzyme replacement therapy, if you previously had been on it, that is an option for you. And the primary objective of this trial is to assess the effect of lucerastat on neuropathic pain. Secondary objectives are really to look at end organ involvement, safety and tolerability, and of course, both cardiac and renal function, and looking at biomarkers. So oral lucerastat monotherapy provides a new mechanism of action to be investigated for the treatment of Fabry's, regardless of the underlying mutation. And it does have the potential, based on the results of the early phase 1b study, of being another possible Fabry disease modifying treatment. It's the first clinical trial that I've been involved with where the outcome measure is neuropathic pain as the primary endpoint. And that, has certainly been somewhat controversial for the different centers involved. But there's also, particularly in the open label extension period, we will be able to develop and gather data on other systems involvement, not just gastrointestinal, but also cardiac and renal. And we'll get much better data in the open label extension to explore long-term safety and other treatment effects. So I need to advise people that recruitment is still ongoing. Because of COVID-19, recruitment was slower than anticipated in this global study, and it is doing much better now than it was. But recruitment is, I think, the target goal of enrolling the number of patients needed to make meaningful conclusions will be achieved by the end of the year. But enrollment will continue and recruitment till December 31st, 2020. And if you want to learn more about this and to see if you're eligible, please look at this Modify website. These are the study sites in Canada. Here I am in Winnipeg, but you can see there are Modify sites throughout Canada. And of course, it's a global study. So there's many countries involved and all working very hard to try to be able to identify whether or not we're dealing with a useful agent for oral monotherapy in Fabry disease. I, as one of the investigators at our site, we do not have any idea which patient has been on in the 6-month period, has been on placebo or on drug. I do know that these data will not be in terms of whether the patient has been on drug or placebo, and the analysis will not be done until all the patients have finished the first 6 months. I know that from a safety profile point of view, there's regular monitoring of the overall data with respect to any worrisome safety problems, but to date, we have not heard about anything. Now, we've had to have many changes because of COVID-19 to the conducting of clinical trials. And I know that the Modify people from Idorsia, all the study monitors, and all the members of our team have worked very hard to try to be able to conduct remote visits to all our patients. And many patients, even now in Winnipeg, really, we're still able to see patients at our research center again, but there's limited face-to-face contact with in most centers around the world because of COVID-19. But people are working very hard, and Idorsia is giving us very good guidance how to complete this study properly. And as soon as we're allowed, other sites are allowed to bring patients on board and onto the research centers, that we do that. So if you are interested and learn more about Modify, please check the website. And I know that Julia will make these slides available for people who want more information. Now, Sanofi Genzyme also has another product of SRT called Venglustat that's under investigation for Fabry disease. I don't have to go over the background again. Again, this is an investigational, another compound that inhibits this GCS enzyme. It does penetrate the brain. It reduces the synthesis of GL1, which is a key precursor of this GL3, which is one of the main offending agents in Fabry disease. Sometimes you see GL3 or GB3, also lyso-GB3. Right now, there has been a phase 2 multi-center open label. That means it's not double blind, a single arm, an uncontrolled 26-week study with a 130-week extension in Fabry's patients who meet eligibility criteria. And this study has actually been completed, and it was to evaluate the safety and pharmacodynamics and the efficacy of Venglustat. This is given once daily. And this initial phase 2 study was only for adult male patients diagnosed with Fabry disease. And these slides kindly provided to me by Sanofi Genzyme this study has been completed, and an additional next study is in the planning stages, but I have no information as yet when to expect that study may be possible. Again, this endpoint was based on the clearance of this accumulating substrate GL3 from the skin endothelial cells in the capillaries. And so there was quite invasive monitoring, and it was looking by light microscopy, and the samples were scored for storage by 3 independent pathologists. Another primary endpoint of the open label extension is safety, looking at adverse events and many of the standard parameters that we monitor in other clinical trials with Fabry disease. And there's other endpoints as well. In this particular study that's been published, stage 2 of Venglustat and Fabry disease, the diagnosis had to be classic. Fabry is confirmed by genotype and residual enzyme activity. The plasma lyso-GB3 level had to be very high, and there was no prior Fabry disease specific treatment. And exclusion criteria, you basically had to have no renal problems and no clinically significant disease. And so this is a study that it was really designed to have initial look at young classic Fabry males with low disease burden. And they were young, and they were basically healthy. And the outcome measures, as you know, were pain and storage. And the safety profile, when they analyzed the results for the first from baseline to week 156, was that the safety profile again was excellent. There were some treatment emergent adverse events. That's not unexpected in most clinical trials where you have meticulous recording and monitoring of patients. You're going to get lots of little things. We all have headaches. We all can sprain our ankles. Most of them were not considered to be treatment related, and the majority were very mild. There are some that maybe they considered them to be treatment related, significant events, but this is still very, very preliminary. But overall, there were no significant changes with respect to end organ involvement, particularly cardiac, noted in this initial phase 2 study of venglustat. There was significant reduction in the skin GL3 scores at week 156. So here we had more severe storage, and by 156, you can see in the green, some of the patients, their skin did not show any storage of any lipid material at all. But again, as you can see, the numbers are small. But the clearance of the skin GL3, even in a small number, is encouraging. And it says to the company, it says to the investigators that there's good indication to go through the next phase of clinical study, which I hope is in the planning stages now. And you can see in the initial phase 1 study, there was significant reduction in the biomarkers in the GL1 and GL3 and plasma. There's other markers, the lyso-GL3 as well. There were numbers are very small again, but there was improvement in body pain and gastrointestinal scores in the young men who participated in the study. And overall, venglustat was very well tolerated with a good safety profile. Again, the skin reduction of storage material is very encouraging, as is the safety profile. And some of the storage material continued to decline more than what was expected. And maybe there's some other pathways that are being used to clear GL3 from the skin and other organs of Fabry disease. So there's still a lot that needs to be learned about Fabry disease. So I think, Julia, that I'm actually kind of on time. I said for 45 minutes. I've tried to give you an overview of the approach of substrate reduction therapy with 2 oral monotherapy agents being studied, one developed by Idorsia Pharmaceuticals, one by Sanofi Genzyme, and where preliminary results are very encouraging. You can see that many patients who've been on ERT for many, many years might want to choose to try oral therapy as opposed to IV treatment. Some patients on IV treatment develop immune responses that would suggest that, IV enzyme replacement therapy is not producing the ideal results. There's many reasons why some patients would consider oral therapy superior to IV therapy, especially in that the oral monotherapies seem to pass through the blood-brain barrier, whereas large enzyme replacement therapies that have to be given intravenously are too large to pass. I want to send sincere thanks to my colleagues at Idorsia Pharmaceuticals who are conducting lucerastat trial. They provided me with the slides and a very, very helpful discussion, and I'm indebted to them. And also to Sanofi Genzyme to give me the preliminary data from the phase 2A venglustat trial in Fabry disease. And I thank Dr. Patrick Deegan, who's the PI for that venglustat trial in Fabry from the UK, who provided me with some of his slides, a very helpful discussion, as well as Dr. Robert Hopkin from Cincinnati Children's Medical Center, who follows a lot of Fabry's patients, who I've had some very good talks with. I'm very indebted to my Winnipeg team, who are absolutely dedicated to this study, all members of the research team, and especially, of course, to the patients and the families, because without them, we wouldn't have any clinical trials. And I know it's a huge commitment to take part in a clinical trial. But I see many patients who want to participate in clinical trials, but who can't, for whatever reason. That it's a tremendous obligation and commitment to take part in a clinical trial, and that's okay. But we need all hands on board, the physicians, the researchers, the pharmaceutical companies, and especially the patients, to be able to make progress in us overcoming the deleterious effects of Fabry disease. And the progress is great. And again, I'm very grateful to the home that I've had in Winnipeg, at the Research Institute and University for so many years. So I'll stop here, and I'll be happy to take any questions. Thank you.
Yeah, thank you very much. You know what, it's a bit of a complex mode of action to explain, and thank you for doing such a good job. I think it's I like those slides that you use. Those are really great in the sense of explaining how we're just reducing the substrate to build up in the first place. I think that's a great way of saying it, because I think that for so many years we've been so accustomed to ERT, which is replacing that therapy.
Replacing enzyme.
Replacing the enzyme. Yeah. So we've had a couple questions come up, and one of them.
Again, I'm sorry again about the slides and the connectivity here, but
no, that's okay. No problem at all. We didn't lose anybody. Everyone stayed with us, so thank you for that.
Thank you.
So one of the questions was, and I know that you touched on this, but it's just, it came up. So it was just the question of, they're wondering, there's no mutation that's immutable. So I think that the patient's obviously thinking of, Galafold. So just to clarify again. Yep. So just to clarify again, it's for
for the chaperone therapy, you have to have an amenable mutation. But for substrate reduction, just reducing the substrate, any mutation in causing Fabry disease, it's not a contraindication. Every mutation, as long as you have Fabry disease and a reduced amount of enzyme, then if we can reduce the substrate somehow, then you're eligible.
Perfect. Thank you. Someone was just asking, two questions. What does open label mean, and the single arm?
Okay. Well, in the double-blind trial, as I say, neither the investigator nor the patient knows whether he or she is receiving placebo or drug. After that 6 months, this is talking about the modified trial, open label extension, that the patient has a choice to continue on the actual drug, knowing that he or she is getting lucerastat. So that is what open label extension. In the phase 2A for Benglustat, they also had quite a long open label extension, but that was not a double-blind trial. That was an open label, I guess, single arm. I'm not quite sure what that single arm meant. They were just giving one drug, the Benglustat, and it was at one dose level. They weren't combining it with anything else.
Perfect. Thank you. No, that's great. Would therapies ever be paired? So I think they're asking, would this be ever paired with gene therapy? Would SRT also be, combined with chaperone therapy?
That's a good question. Very good question. I see in the initial studies of Benglustat, that very first one, no, in the initial studies in the modify, I think the very, very preliminary studies, it was an add-on to the enzyme replacement therapy. And I don't see why, a protocol could not be developed where it could be used in combination with chaperone therapy. I don't see any contraindication for that, but I haven't heard of any of those ones in the planning. The main thing is will all these ultimately be replaced by gene therapy? And, that is the hope, that if you can imagine, you get one injection of a gene that gets incorporated in all your cells that begins to produce normal enzyme. Will that be the cure for the disease? And will that be for everybody? Probably not. I don't know where gene therapy will lead us in Fabry disease. I think it is, the trials are underway, And I know Dr. Khan in Calgary and Dr. West in Halifax are sites, and many people are involved in that. And, encouraging results that even after so many months after gene therapy being infused, there is residual enzyme activity and clearance of biomarkers. But whether or not all patients will be candidates for gene therapy, if you have high antibodies, how are we going to deal with those with gene therapy? I mean, we have those problems with the enzyme replacement therapy trials too.
Yeah.
But I think gene therapy holds tremendous promise.
Yeah, I think so too. Wonderful. We have a comment here, Dr. Greenberg. This is such exciting news for Fabry patients. Very encouraging. Thank you. And a question, can you talk a bit about the risk of a patient stopping ERT and ending up on a placebo for 6 months during the clinical trial?
I know that this was discussed at many, many meetings. The caveat was that, Fabry's is a slowly progressive disease. There was no evidence that anything catastrophic would happen by stopping the enzyme replacement therapy for that period of the 6-week screening and the 6 month of the double-blind trial. I think in that the protocols that were developed by Idorsia, they had input from many people. It was approved through the regulatory bodies, and that there was no evidence that stopping for 6 months and 6 weeks was going to be detrimental. But the caveat also was if something was, but the perk was, of course, that patients both wanted to contribute to research and participate in clinical trials, but also having a holiday from IVs, where IV access is sometimes very difficult, was a real strong motivator for some patients. But if a patient and the patient's doctor felt that there was some deterioration during the double-blind phase, that they could restart enzyme replacement therapy as well. So there was a way that the patient shouldn't feel too jeopardized or too vulnerable if they wanted to participate in the trial. As I say, it's a huge commitment to take part in the trial, and it's many patients naturally have that kind of fear that, it's unproven therapy. That's why you do the research trial. And if something has worked for you, do you really want to quote rock the boat? And we have patients like that who've chosen not to be considered for an SRT trial because they're satisfied on ERT. It's really in many ways a personal choice, but I know that we try to do everything to give people all the information and help them make a decision that's best for them.
Yeah, great answer, and very thorough too, because I think that is something that would sit on people's minds a little bit, but it makes perfect sense. Yeah, I mean that's all the questions that we have. That was an excellent presentation. Thank you so much. There's going to be a lot of people who are going to want to watch this after, so this is wonderful and a great resource for us to be able to share on our website too for anyone that's wondering about SRT. So
Yeah,
again,
as I say, stay safe, everybody. We're all preoccupied and want that COVID vaccine, but I think families with Fabry disease in general are doing well. We follow that literature very closely, but continue to take care. And it's been a very, very amazing time to be able to actually proceed in a such positive way in introducing these new treatments and the treatment clinical trials for Fabry disease. And if we're able to do that during such difficult times, imagine when the COVID's over, just how good it's going to be.
Yeah, that's right. Good point. Yeah, well, thank you so
Okay.
much. And it is very exciting. And just again, thank you for your time tonight.
Oh, no. Oh, it's my pleasure. And I'm sorry again for some of the connectivity problems, but I think in the end, it went okay. Okay.
It went great. It went so good. Thank you.
Okay.
Thank you,
Nice to see you.
everybody.
It's nice to see everybody.
Nice
Take
to see
care.
everybody. Have a
Bye-bye.
great night.
Okay. Bye-bye.
Bye-bye.